Good day, grade 11 life science students. Thank you very much for connecting and tuning on to Metric Hub. I just want to quickly introduce to you and work on the production of carbon dioxide during cellular respiration. Investigative questions, experimental questions are very key. There are also areas and papers which we must get the maximum marks because the information is quite not that much confusing. So we are going to look at an investigation, an experiment question to see if carbon dioxide is produced during cellular respiration. So in this case, obviously, as grade 11 students, I'm aware and I'm so sure that investigative questions, experimental questions want you to understand to distinguish between an experiment set up and the control of the investigation, also being able to make suitable conclusions in a given experimental investigation. There is an issue to do with the validity of an investigation, how to ensure that results that are obtained in an investigation are very correct and are applicable to the experiment that has been done. So what I'm saying is we are going to be talking about all these. I'm sure you have seen such a setup like this one where we use germinating seeds or otherwise some small living organisms to do the experiment. So let's just quickly go to the question. Let's just quickly go to the presentation and we can explore it together. What's happening here is that we want to see if carbon dioxide is produced or released during cellular respiration. So we can only do that by scientific measures or scientific means. So in this case, we have got materials that we use. We have got lime water. Lime water is used as our one. We have got test tubes or views that are also used. Straws or glass tubes, we also use them. Rubber stoppers, which are also going to be explained in the investigation. I said we can as well use germinating seeds or a small animal in terms of assessing or having them as a source of carbon dioxide production. We have got the jars or beakers, of course, they are supposed to be used. Optional of we have got optionals which can also be soda lime that is used to absorb carbon dioxide. Number 6, we have got also potassium hydroxide that can also be used to absorb carbon dioxide as well. And then we also gonna use water bath for temperature control. So these are the materials or these are the requirements that can be used or that we need when we are to do an investigation on production of carbon dioxide during cellular respiration. So I quickly want us to look at the procedure, what needs to be done. We have got one, set up the control. I guess you know what we mean by the control. A control in an investigation. What is the importance of a control? What's the importance of a control in an investigation? First, we are saying fill one test tube with lime water and seal it with a stopper and a straw. This will serve as a control showing how lime water reacts with what? With air alone. So here we are saying we want to see that the lime water reacts with the lime water reacts with air alone. It means there is no carbon dioxide in this air. So a control, what is happening there is that it is used, it's used to uh, compare the results in an investigation. In an investigation, a control is used to compare the results. So what we mean is when we are saying compare the results, we are able to tell that carbon dioxide is produced during cellular respiration by having a setup which is having a natural setup, which is having the best, or which is having the conditions that are unmanipulated. So what we are saying is that this setup, the control is very important. When an investigation is to be done, we always supposed to have a control. We can say that it is used as a benchmark in terms of making conclusive scientific findings in an investigation. So we have got prepared the experimental setup. The experimental setup, we are saying this one, it is the one which is always supposed to be, it is the one which is manipulated. It's the one that is used to do the investigation. It's used in the investigation. So few another test tube with lime water, place germinating seeds there, place germinating seeds in the lime water. I mean, place the germinating seeds, uh, and then or a small animal. We are saying other times you shall see, whereby you are asked to say, give an alternative of germinating seeds in this investigation. Instead of using germinating seeds, what's the alternative, or what's the option that can be used? It can be a small animal. So we are saying both are used as sources of energy, I mean sources of carbon dioxide. For we are saying it's a source, is a source of what? As a source of carbon dioxide. As a source of carbon dioxide, that is why we need the germinating seeds, or what? Or the small animal. So with a small amount of water to keep them moist, we are saying with a small amount of water to keep them moist, small amount of water to keep them moist. This is for seeds, or in a separate container with perforated partition for the animal, allowing for air circulation. In any case, what you need to understand here is that when we are doing an investigation class, it is very important, and it is a requirement that no organism must be affected. It must not die, or it must not it must not die. It must not die, or it must not be affected by the investigations that are to be done. That's one thing that you should know. So we are saying the small seeds, we are also putting some amount of water to keep them moist. So therefore we are giving them the conditions that might also cause germination. We are making them moist in order for germination to take place. And also, if it is a small animal that is to be used, we are saying it must have also some holes. There is a separate container that must be in there with some holes, which is allowing for air circulation. That is one thing that you should also understand. So step number 3, we are saying introduce carbon dioxide. For seeds, seal the container holding the seeds. Seal the container, we are saying seal the container holding the seeds and lime water. Ensure the straw or glass tube is submerged in the lime water. It means to say it is sinking. It is sinking in the lime water. That is that the straw or the tube is sinking in the lime water. This container holding the seeds must sink in the lime water. And then for small animals, seal the container with the animal. You seal the container with the animal, and soda lime if you are using it, and the straw and the glass tube submerged in lime water. So this is one step 3 now, that we must ensure that the animal also, it is actually we are saying we must seal the container with the animal and soda lime. Remember, we said soda lime is used to what? To absorb the carbon dioxide, and the straw also, so therefore they submerge in lime water. These are also, that is step number 3. We are talking about an experiment to determine if carbon dioxide is produced what during cellular respiration. So incubate, step number 4, incubate. Uh, in this case, place both setups in a dark room or a warm place for several hours for overnight. The question that I want you to understand is why should we put uh, these in a warm place for several hours or overnight? It is to control the germination conditions. We need to control the germination conditions, control the germination conditions and ensure consistency of results. We are saying we are to understand that there must be an incubation which must be done by placing both setups in a dark room, in a dark place, warm place for several hours in order to control the germination conditions and ensure consistency of results. So we are presetting conditions for germination. And then number five here, examine the lime water in both setups. Examine the lime water in both setup. So the lime water in the control setup should remain clear. The lime water in the control setup, which we said that there is no germinating seeds there, it remains clear. Well, the lime water in the setup with a germinating seed or animal should turn milk cloud. I'm sure you are seeing some differences here. We are saying lime water in the control setup remains clear. Therefore, I don't want to say something right now, but the one with germinating seeds or animal turns milky or cloud. What's the explanation for that? What's the explanation? The cloudiness of the lime water indicates the presence of carbon dioxide. So in other words, we are saying lime water, it is used to test, it is used to test for, it is used to test for presence of carbon dioxide. Lime water is used to test for presence of carbon dioxide, and what happens? It changes to milky or cloudy. It changes to become cloudy or what or milky. So germinating seeds and animal respire producing carbon dioxide as a byproduct. I'm sure now we can see what we are seeing is carbon dioxide produced during germination, I mean during cellular respiration. So we are saying yes, because when animals and germinating seeds respire, their byproduct is what is carbon dioxide. This carbon dioxide reacts with lime water, forming calcium carbonate, a precipitate that makes the solution cloudy. It forms a forming calcium carbonate, which makes the solution cloud. In this case, that's what happens there. So let's have the additional note. So in as a conclusion, carbon dioxide is produced during cellular respiration, and it can be proved by testing it with lime water, which gives the color, which gives the changes that we are giving there. So additional notes that I want you to take note of: soda lime is used to absorb carbon dioxide. That is one thing that you need to take note of. In some setup to demonstrate that the observed change in lime water is due to what? To respiration, just not from what? From air exposure. So now, understand this class. Soda lime is used to absorb carbon dioxide in some setup to demonstrate that what? The observed changes in the lime water is due to what? Is due to respiration, not just from air exposure. So when we say air exposure, we are talking about atmospheric air. That's one thing that we should remember there. And then number two, respirometer can be used to quantify the amount of oxygen consumed during cellular respiration by measuring the change in gas volume. So what is happening is respirometer, it is it is an instrument that can be used to actually that can all actually be used to quantify, we are saying to measure the amount of oxygen consumed during cellular respiration. This, I'm sure, remember that during cellular respiration, oxygen is used. Oxygen is used during cellular respiration. So we can be able to know the amount of oxygen used by a respirometer. So therefore, we can see the change in gas volume that is there. So uh, Prothrombin blue can also be used as a pH indicator, can also be used. So in terms of measuring this presence of carbon dioxide, we can also use the Prothrombin blue, which is BTB. It can also be used as a pH indicator to detect carbon dioxide production. So carbon dioxide production can be tested by this is a second option, which is Prothrombin blue, which we are saying it is used as a pH indicator. So this is just a chemical compound that changes color depending on the pH of the solution that it is in. So the potential hydrogen atoms that are around it is used to determine, it is used also to change, to confirm the presence of carbon dioxide. You shall see this. So what we are saying is that this chemical compound changes color depending on the pH of the what of the solution that it is exposed to. So check here. Uh, BTB is used to indicate carbon dioxide production during cellular respiration because because it is a pH indicator. So it measures the pH that changes color on the acidity, based on the acidity class or alkalinity of a solution. I'm sure that we understand that pH is measured. It is a measurement of potential hydrogen atoms which can actually be seen by the acidity or alkalinity of a solution, or it determines that. So when carbon dioxide dissolves in water, it forms carbonic acid. Carbon dioxide and water, when it reacts with water, we are saying it dissolves and then forms carbonic acid, which lowers the pH of the solution. So it lowers the pH if carbon dioxide dissolves in water and making it more acidic. So when pH is low, the I mean when the pH of a solution is low, it means also it is acidic. So the BTB turns yellow in acidity conditions class. It turns yellow in acidity conditions, which means in lower pH, and blue in basic conditions where the pH is high. So uh, the prothrombin blue can also be used to determine presence of carbon dioxide. So it turns to yellow. That is when the carbon dioxide is there, which shows that there is acidic conditions. But if conditions are normal or high pH, we have got, I mean, the BTB, it maintains a blue color. So therefore, by observing the color change of this BTB that we are talking about, when we check the color change, it can qualitatively assess that the amount of carbon dioxide produced during cellular respiration uh, as increased. Carbon dioxide production leads to what? To acidity conditions, and color shift to what? To yellow. So this is another way that can be used, class, to determine carbon dioxide production during cellular respiration by using the bottom blue, which changes to what? Which changes from blue to yellow if carbon dioxide is produced. So we can qualitatively assess the amount of carbon dioxide produced during cellular respiration, because the increase in carbon dioxide production, it leads or it causes the solution to become acidic, which is noted by the color changes that we are talking about in this regard. So That is one thing that you should understand. So I'm having this just to comprehensively conclude on carbon dioxide production during cellular respiration. We have got cotton wool. In some cases, examiners, they would want to ask you, this can also be used to, to maintain temperature, to maintain temperature, so that we can also cause germination and to maintain temperature, so therefore we can cause germination, and then that is the and also the advantage of what, of moisture also, which is supposed to be there. And then we are saying we have got lime, clear lime water that is given there. So now what happens is that, give one reason why the seeds were sterilized before they were used. So during this experiment, seeds are sterilized. They are sterilized. The reason is that we want to ensure all microorganisms are killed. This is to ensure that the microorganisms that might penetrate or that might have borrowed through the seeds are dead. So the seeds are sterilized so as to ensure that. That is one thing that you should understand. State one way how the learners could ensure the validity of the investigation. So validity of investigation, we are saying, we are talking about reliability, how the learners can ensure reliability of the results. Reliability of results. How can it be improved? So we are saying, one, using the same type of seeds. Same type of seeds must be used. They must use same type of seeds, same amount of lime water, same size of test tubes. Repeating the investigation. They must also repeat the investigation. Repeat the investigation. Are we together here, class? Repeating the investigation, it can also be one way of improving reliability of results. We can also ensure that the readings are done by one person. Checking and recording of results is done by one person. Is done by one person. That can also help in ensuring that the results are correct. That's what we need. That can also be done to ensure that the results are correct. And also, even in this case, uh, having a bigger sample size of seeds, using a big sample size. The sample size, we are saying, in just ensuring that the seeds are many enough, so therefore we can easily be able to see the production of the carbon dioxide that we are to investigate. So that's one other thing. So state suitable conclusion for the investigation above. So this investigation above, class, it's about investigating the production of carbon dioxide. So clear lime water turns milky in presence of carbon dioxide. Clear lime water turns milky in presence of what? Carbon dioxide. So that is the conclusion that can be taken, that can be given in this regard. We are saying this clear lime water, it turns milky in presence of carbon dioxide. That is one thing that we need to understand. In presence of carbon dioxide, the clear lime water turns milky, and then that is very correct. And then explain how they would set up a control for this investigation as a question. Also, in this case, how can they set up and control? The apparatus would be set up in the same exact way. set up the apparatus in the same exact way by using no seeds or seeds that were boiled to ensure that changes observed in the investigation were caused by cellular respiration or germinating seeds. That is one way that can be done. So we are saying a control of the investigation must be set up in order for us to compare the results. So therefore, the results that are to be made are attributed or can be conclusively said that they are due to what? To production of carbon dioxide other than any other factors that might affect the experiment. So we can also do, I have got another way also, which is procedure 2, which can be used in terms of determining the production of carbon dioxide during cellular respiration. So we might have this setup class as what? As another setup that you can see initial level and then water level, final level. If you can see, so you are saying level 1 and then we have got level 2 in terms of the level of water. And then we are having a bent glass tube that is actually connected to the germinating seeds here. It is connected to the germinating seeds, and then potassium solution here, we are saying it is also potassium hydroxide is also used as to absorb carbon dioxide, to absorb carbon dioxide. That is what is happening with the potassium hydroxide. And then we have got the germinating seeds. So take 30 to 40 seeds of gram soaked in water and allow them to germinate by placing them in moist cotton wool or a blotting paper or a moist cloth for 2 days. So germination must be allowed. Transfer the germinating seeds with whitish outgrowth into a conical flask. A conical flask, as you can see here, this is a conical flask that we are talking about. The conical flask, when they are about to germinate, they are transferred into a conical flask and add a few drops of water to keep the seeds moist. We are saying some water is added to keep the seeds moist. Take the freshly prepared solution of 20% potassium hydroxide, 20% of potassium hydroxide in a test tube and suspend it in the conical flask using a thread just above the germinating seeds. This is actually what we are seeing here. The potassium hydroxide here is given. It is suspended on top or above the germinating seeds with a thread there. So that is what is happening with point number 3. And then the mouth of the conical flask is closed by placing the rubber corks with one hole, and then ensure that the thread tied to potassium hydroxide is firmly placed and held correct at a correct distance. As you can see, we are saying at a distance that is enough from the germinating seeds. And then take the U-shaped delivery tube, insert one of the ends in the conical flask through the hole in the rubber cork. The other end to the delivery tube is placed in a beaker containing water. Are we seeing this class? We are saying a conical tube is, it is contained. This is what is happening. It is connected through the cork to the conical flask and then to the container containing water. And then that is what we are talking about, the bent tube. It is connected to this. And then what happens next? We are saying seal the connections of the experimental setup with Vaseline. Vaseline is used to make the apparatus airtight to avoid. Why is Vaseline applied? Why Vaseline? Why? To make sure that the apparatus are airtight to avoid air to enter or leave the conical flask. Mark the initial level of water in the delivery tube and leave the setup undisturbed for 1 to 2 hours. Observe the change in the level of water in the delivery tube immersed in water inside the beaker. This is what we can see. This is what we can see here to say we are saying the conical bent tube is connected from the germinate from the conical flask, and then we are saying the reading number one reading, we are saying reading one, and then we have got our reading two. So this is before, at the beginning of the experiment, and then we are saying after the experiment. After the experiment, then what happens? So what we are saying is that they mark the level of the water before and after. So we are saying the final level of water is shown there. That is what happens there. So let's see, and then what observations can we make? After 2 hours, it can be observed that the level of water, it rises up in the U-shaped delivery tube dipped in the beaker from the initial mark level in the diagram above. I'm sure that we can confirm together to say that the level of water, it rises up in the U-shaped delivery tube from the initial thing. What we are seeing is that the level of the water increases, as I've seen here to say we have got reading one, reading two. So at the beginning it's less or it's lower, and then it rises to that level that is shown in the second thing. So germinating seeds in the conical flask releases carbon dioxide during cellular respiration, and which is absorbed by potassium hydroxide kept in small tube there. And then due to absorption of carbon dioxide, a vacuum is created in the flask, which causes a rise in the what? Which causes the rise in level of water in the delivery tube. So due to absorption of carbon dioxide in the vacuum, in the flask, we have got a cause in rise in the level of water in the delivery tube. So now a vacuum is created there. I want you to see this thing. We are saying the absorption of carbon dioxide here, it creates a vacuum. When carbon dioxide is absorbed by the potassium hydroxide here, we are saying a vacuum is created in the bent glass here. So therefore, what happens is that the space, the level of water, our level of water, which is on 1, rises to a level which is maybe 3 there. So the ability of the water to rise is because of a vacuum that is created in this bent glass. The bent glass, we are saying there is a vacuum that is created. A vacuum is created here, and then what happens with the vacuum? It causes now the level of water to rise. So because the carbon dioxide is being absorbed by what? By the potassium hydroxide there. That is what is happening. So in this case, the absorption of carbon dioxide, uh, due to the absorption of carbon dioxide, a vacuum is created, and then in the flask, which cause the rise in the what? In the level of water in the delivery tube. So what is happening is that carbon dioxide is what is produced, which causes a vacuum to be created in the conical flask, and then level of water rises in the delivery tube, which we have seen there. So in other words, this is a setup class that you might also see, and you need to be able to relate and explain it very well. Thank you very much for your time, and I hope that you have understood and you have seen quite an interesting experiment that you can as well you might be asked in the exam, and we have set up and explained this. So how to increase validity of the results? How can we ensure that carbon dioxide is absorbed and so forth in this case, as well as what is the control and how to set up a control in an experiment. So thank you very much for your time, and all the best in the upcoming end of term exams, and let's keep connected by turning on your notification button share and like the videos. You are to ensure yourself that finally no repeats, but progress continues. All the best. Let's meet again in the upcoming videos and keep connected. Share to friends and family. Let's pass together and let's have one spirit of determination towards education. Have a good day.